Objective: to study about the opening and closing of stomata. Opening and closing of stomata. The immediate cause of opening or closing of the stomata is a change in the turgidity of the guard cells. The inner wall of each guard cell towards the stomatal aperture is thick and elastic. When turgidity increases within the two guard cells flanking each stomatal aperture or pore, the thin outer walls bulge out and force the inner walls into a crescent shape. The opening of the stoma is also aided by the orientation of the microfibrils in the cell walls of the guard cells. In the guard cells, cellulose microfibrils are oriented radially rather than longitudinally. This makes it easier for the stoma to open. When the guard cells lose turgor due to water loss or water stress, the elastic inner walls regain their original shape. The guard cells become flaccid and the stoma closes. Mechanism of opening and closing of photoactive stomata. The K+ pump theory or active proton concept was proposed by Leavitt in 1974. Opening mechanism. In the presence of sunlight, the starch already present in the guard cells is converted into malic acid. Malic acid dissociates into malate and H+ ions. The outward movement of H+ ions from the guard cells requires metabolic energy in the form of ATP, so it is called active transport, whereas the movement of K+ ions occurs in exchange of H+ ions. According to this theory, accumulation of K+ ions into the guard cells from the subsidiary cells occurs in the presence of light. As K+ ions migrate into guard cells, in excess of H+ ion exchange, they are suitably balanced by the intake of Cl ions. The two anions malate and chloride combine with K+ ions in the guard cells, resulting in potassium malate and potassium chloride, respectively. These are osmotically active. These salts increase the concentration of guard cells, thus decreasing the water potential. Delta phi W value becomes more negative. Therefore, water potential gradient minus delta phi W is established between the guard cells and their surrounding epidermal cells. Due to the water potential gradients, water moves from the surrounding epidermal cells with higher water potential, with higher delta phi W, into the guard cells with lower water potential, lower delta phi W. It results in increased turgidity due to endosmosis. This endosmosis into the guard cells makes the outer wall more convex and drawing the inner walls apart and thus finally opens the pore. In monocots, when the guard cells expand by water intake, their ends with thin walls bulge outwards and draw the middle thick walls apart. This results in stomatal opening. Closure mechanism. In the dark, K+ uptake is inhibited by CO2 and ABA by changing the membrane permeability. The K+ and Cl ions move passively out of the guard cells into the surrounding epidermal cells. The guard cells also dispose of the accumulated malate, partly by consumption during Krebs cycle of respiration and partly by releasing it into other epidermal cells. The efflux results in the loss of turgidity within the guard cells due to exosmosis. The thick inner walls of the guard cells return to their original position, resulting in the closure of stomatal aperture.